Hi everybody, I am just going to talk to you guys a little bit about our new unit that we're starting on the diversity of life. And this flip lecture, you guys are going to need to take notes on it in your composition book. And it's going to lead into a little Candy lab activity that's going to be a great introduction for us to better understand how to organize all the diversity of life that we have on the earth. So the learning targets for this particular lesson are listed here. And it says here to analyze classification systems created by scientists that organize the diversity of organisms and show their degree of evolutionary relatedness. So if you can imagine, there's all kinds of plants and animals and bacteria and this and that. And to organize all of them, they have to look at a lot of characteristics, not only external, like phenotypic characteristics, but also molecular. And then that helps them decide on their degree of relatedness with each other. And also, we need to be able to construct a cladogram. And a cladogram is basically the actual tree that you would make as a result of looking at characteristics between organisms. So as you see here, 7.1, it says to analyze the classification of organisms according to their evolutionary relationships, including cladograms. So on the end of course exam for biology, we're definitely going to have to know how to construct a cladogram. So this activity is going to be a little intro to this entire unit. So the first thing I'd like to do is talk to you about what phylogeny is and what cladograms are. So phylogeny is when scientists look at the evolutionary relationships that exist between species, and they usually represent them in something that they call a phylogenetic tree. And the actual tree is what we call a cladogram. So what this does is it attempts to answer the questions of the relatedness or relationships we even have between species. And the trees are constructed using a cladogram, which is pictured here. Okay. So as you can see here, there's three main groups. There's bacteria, there's archaebacteria, and then there's eukaryote. These two here are two main areas of bacteria. So there's regular bacteria, which is called eubacteria, and that is the bacteria that we're probably most familiar with. So E. coli is an example of that. Archaebacteria, on the other hand, is ancient bacteria, and they live in like really extreme conditions. So there's like one type that's called an extreme halophile, which is a type of bacteria that can live in really, really salty conditions and water. And there's also thermoacidophiles, which are bacteria that can live in hot sulfur springs, like at Yosemite. So those types of bacteria are really ancient and exhibit a lot of the characteristics that they believe the early forms of life had. And as you can see, it wasn't just one species here, right here. It actually branched out into other species. And those other groups are in their own grouping because of their unique characteristics that they have. The eukaryotes are organisms that have a cell plan that has organelles on the inside. Bacteria have the opposite, which is prokaryote. So prokaryote, hence the word prefix pro, means like first. These are, it's a type of cell plan where there's no organelles on the inside and no membrane-bound nucleus, whereas eukaryotes, which is what we aren't just one cell, though. We actually have many eukaryotic cells. We have those as well as dogs, manatees, plants, and so on. You can see it all right in here. So there's a lot of areas here that we could focus on. Obviously, the phylogenetic tree is much more detailed than this, and that will be in the presentation that Mr. Keller goes over with you. Okay. So what I want you to understand here is the fact that there's lots of life on Earth, and we have to organize it, and we organize it through the use of phylogenetic relationships, and we illustrate it through the use of a cladogram. So the cladogram is organized in a way where it looks at degrees of similarity between groups of organisms. Now these degrees of similarity, the characteristics they could focus on, could be molecular, it could be phenotypic, it could be anatomical, it could be a combination of all of those. And basically what it aims to consider is all the characteristics that they can find. Today you will start a chocolate bar candy lab, and you will be organizing candy bars based on their degree of relatedness. And you'll classify the candy bars into different groupings. You know how like some candy bars are made of milk chocolate or dark chocolate. Some have like a nougat center. Some have nuts. Some don't have nuts. Some have coconut. Some don't. And so you're going to be classifying the candy bars based on as many characteristics as you can find in the candy bars. So this is going to be a nice little introduction to this whole unit because I'm not going to tell you how to do it. I want you to try to figure it out on your own, but I am going to give you a little hint as to how you should start it, which leads us to this last slide. So what we're going to look at is the phylogeny of candy bars, and as you can see here in this picture, this group of students organized these candy bars based off of like dark chocolate or milk chocolate, and then it branched off into like if it had a candy coating or not. It even includes if it has caramel in it or nuts and so on. So to the best of your ability, you and your lab group will classify the candy bars that you'll get in a little Ziploc bag. You're going to use this image as a guide to do it. And in addition to your baggie of chocolate bars, you're also going to get a white paper. And on that white paper, I want you to create a cladogram, okay? And I want you to keep in mind that at the base of the cladogram is what all of them have in common. So you could almost guarantee that they're all going to have chocolate, right? So that would be like your just basic chocolate candy bar. And then from there, it branches off into many different groups. And you'll also have markers in the classroom. One thing to keep in mind too is that the candy bars that are listed here, that are shown here, are not necessarily the ones I'm going to give you. There's going to be different ones. And you also need to keep in mind that you may need to open your candy bars. Do not eat them. And you're going to need to slice them open so that you can make further observations, because there may be some things in it that you don't know. You may want to consider looking at the ingredients in it to get very specific, okay? If you don't finish this today, you need to put all of your materials on a lunch tray that's provided, and we will continue it tomorrow as a lead-in to Mr. Keller's beginning of this whole unit, okay? So we're going to, if you don't finish it today, we'll finish it at the beginning of class period tomorrow, and then we will have a little brief discussion on what you guys found and look at everybody's different ways of organizing it, and then we will start the unit, okay? All right, that's it.